Okay, now the idea here, if you look at the change, the change here, you realize that we start with the PBr322 plasmid, PBr322, which consists of 2 antibiotic resistant genes. Okay, 2 antibiotic resistant genes, and then we make a newer version, PUC19, which only consists of 1 antibiotic resistant gene. Okay, plus Lexac. And then now we have the Pgro. Okay, the newer versions of the plasmids, Pgro, also contain 1 antibiotic resistant gene. Plus green fluorescent protein. So the main idea why we want to change from PBr322 to PUC19 to Pgro, basically you have to know that we want to reduce the number of the antibiotic resistant gene. We want to reduce because there is a risk if this antibiotic resistant gene spreads to the pathogenic bacteria, then particular bacteria will develop the antibiotic resistant strength. So in this case, we want to reduce. At least in this case, you look at the PBr322 ampicillin resistant. They have both, right? Tetracycline resistant. So it means that ampicillin and tetracycline won't be able to kill both. This bacterial, but if PUC19, they only resistant to ampicillins, but we still can kill it by using the tetracycline. Can you see that? Same goes to the Pgro. Okay, so we want to reduce the risk, therefore we change. Okay, we want to reduce the use of the antibiotic resistant genes in the screening process. That's the main idea why we want to actually change from PBr322 plasmid to plasmid PUC19 and then Pgro. So today we're going to look at what's so special about green fluorescent proteins, okay, and how we use this green fluorescent protein in gene technology, okay. So first of all, if you look at the structure of these green fluorescent proteins, it's basically from something a barrel, okay, you can see that something a barrel. So this barrel actually surrounding one of these very important component inside we call as a chromophore. Chromophore is the one that gives us the signal of this green fluorescence. Okay, can you see that it's a barrel, something like cylinder, something like this, a cylinder. So this cylinder, something like protect, okay, the chromophore. So chromophore actually inside in between or inside this cylinder, okay, this chromophore. So what's so important about this green fluorescent protein? Now, before we start for the use of green fluorescent protein, I give you guys one scenario here. Okay, so now look at this. If you want to use it in animal study, how you make sure that the gene have been successfully taken up by this animal. For example, if you look at this, okay, if you want to let's say sometimes we want to incorporate a particular genes. Let's say, for example, certain gene will cause the disease. Let's say, for example, in this case, it causes the disease of the eyes. Okay, eye disease. Okay, so what we're going to do here is we want to actually incorporate this gene in the mice so that the mice develop the disease. Then we can try on our drugs. Correct or not? Can you imagine that? We want to give, for example, we want to use a gene therapy. We want to actually inactivate a particular gene. So how we know? So if without the green fluorescent protein, what we do? Without green fluorescent protein, then we're going to do a random test. So out of 100 mice, let's say I have, okay, I'm going to choose maybe 10 of them, and then I kill them. Okay, kill them, take out the cells of the eyes, and then we check, okay, whether we check whether they develop the disease or not, whether they take up the gene of the interest or not that we want to study. Can I see that? Are you clear? So in this process, we call it invasive process. Means that we have to kill the mice, and also the rest, the remaining 90, how you make sure that they already take up the gene of interest. Correct or not? You just based on the probability. Oh, let's say I take up 10. 9 of them develop, so 90% already developed. So means that your sample also won't tell us directly how accurate this or how this the mice have taken up the gene of interest, right? But with the green fluorescent protein, what we can do here is we can tag our gene of interest. our gene of interest, we tag it along with the green fluorescent protein gene, GFP. Okay, and they share the same promoter. So it means that if green fluorescent protein is expressed, the gene of interest will be expressed as well. Can I see that? So it's indications that we will be able to, or this gene already been taken up. So in this case, you shine with UV, you will realize that, oh yeah, mice number 1 and mice number 3, they already successfully taken out the gene of interest. So mice number 2, no. So we exclude mice number 2 from our experiment because mice number 2 didn't take up the gene of interest, means they won't develop the eye disease. Can you get the idea here? So why we want to use this green fluorescent proteins? Okay, we just shine them with the UV lights, then we will be able to select which mice have been successfully taken up the gene of interest, they will develop the disease, then we can try our drugs onto it. But if you include number 2 in your experiment, they don't have any disease, right? So it means that you use the drug also useless. So your experiment's results is not reliable. Can I say that? Okay, so compare the previous without the green fluorescent protein and now with the green fluorescent protein. So that is advancement in terms of the research here. Okay, so we want to look at the property of the green fluorescent protein, why this green fluorescent protein make them suitable in terms of the research, and what are the limitations as well. Okay, now you do not need to remember all of them. I will highlight some that you need to know. Okay, so what are the advantages? Okay, what are the advantages of using green fluorescent protein in recombinant DNA technology? So first, green fluorescent protein is exceptional powerful tools in recombinant technology because expressing cells simply grow green. It will give us the green color, the green fluorescence, and can be viewed under the conventional fluorescent microscope. So it means that we do not need to kill our cells, right? If let's say you want to know how the cell express, then you have to kill the cell and then extract the protein, then we only know whether they successfully transform or not. But now, very simple, you just view it under the light microscope, okay, sorry, not this fluorescent light microscope, then you will be able to see which cell actually take up the gene of interest already, okay? And very important also, the light emissions from the green fluorescent protein does not require any cell fixation or cell permeabilizations and cell lysis. So we say that no invasions or minimal invasion. You do not need to kill your cells to check whether, okay, the cells actually take up the gene of interest or not, okay? And the barrel light structure that protect the chromophore is very stable, resisting the temperature up to 65 degrees Celsius, and ionic detergent give high resistance to the proteases. Now, this is very important also. Some protein that express in the cells will be degraded by the proteases. Now, what will happen? Let's try to imagine, sir. So we want to study time point. We want to have the time point study. Now, what's mean time point study? So if I want to check the expression every hour, how much expression, right? Okay, so I can plot the graph. Okay, intensity of the green fluorescent protein versus the time. So you can see that there is increase. Correct? Let's say, for example, increase. Okay, but the question here is, if this green fluorescent protein is non-stable, if I say if, and it can be degraded by protease. Protease means the enzyme that cleave the protein. Then what will happen? You won't be able to use it to study the time point. Can I see that? You won't be able, because one is expressed, it degraded by the protease, continue to express, degraded. So means forever you won't get the representative graph here. Are you clear? Okay, so that is the idea of number 3. And number 4, the fluorescent emission does not bleach when excited with a UV light. Some these fluorescent will be bleached. Basically means that when shine with your UV lights over a long period of time, the color intensity become lower and lower. But the fluorescence emissions by these green fluorescent proteins does not bleach. Okay, and high level of GFP expression have not been seen associated with any cellular toxicity. So basically means that if we want to study a time point, we don't need to worry, high concentration of the green fluorescent protein going to kill our cells. No, okay? And number 6 here, the barrel-like structure of GFP also allowed numerous N-terminal and C-terminal fusion genes to be made and expressed without the loss of the fluorescence activity. And consequently, GFP can be used to study the behavior of tag protein within the living cells. Now, what does this mean? Number 6 here, let me show you something a bit special about this green fluorescent protein. Okay, a green fluorescent protein. Now, what's so special about this green fluorescent protein? We can do the gene fusion. And these gene fusions allow us to have the fusions of the protein. Okay, protein fusion. Now, what does this mean? So far, not many proteins can do this, what we call the gene fusions and also the protein fusion. If you do the protein fusion, basically look at this. If I want to study this gene of interest, this is my gene of interest, I can fuse it, okay, by the DNA technology, making this what we call this sticky end. Then we can join the green fluorescent protein in front of them. So this green fluorescent protein, and then this one is the gene of interest. So with GFP, what will happen here is GFP will code for the green fluorescent protein, the barrel-like structure with the chromophore, and then the gene of interest also will code for the protein attached to this green fluorescent protein. And surprisingly, this gene of interest code for the protein, and this protein remain the same. So means that they still function well. Are you clear? Can you get what I mean? We can do the protein fusion, fuse the protein together with the GFP. So what's so important about this? What's so important about this? Basically, if I shine with UV and I get the emissions of the green fluorescent signal, so indicate what? Indicates that my proteins can function. Cannot, because they're attached together and proteins can function. But if you fuse our gene of interest with other gene, let's say I fuse with gene of interest, with, let's say, simply any other gene. Okay, so if this red color gene alone, maybe you form the protein like this. Okay, blue color gene alone, it may form this. But when you fuse them together, what will happen is the blue color and green color may form a different type of protein, which both of them lost the functions. You cannot simply fuse the two genes together like this, because the protein, the products here will be wrong. It will be spoke. But this phenomenon is not seen in the green fluorescent protein. So making green fluorescent protein still function, give us a green fluorescence, and the protein of interest still remain functional. So in this case, if I see the cells, if I view it under this UV, I see the cell give us the green fluorescent, I know that the proteins that we want the cell to express, to form, is there. And the proteins still carry out the function. So making this green fluorescent protein a very, very suitable proteins to be used to study, okay, to study these expressions of the genes in a particular cells. Okay, yeah. So come back to this notes here. So which out of 6 points here, so far no AC question will ask you, okay, why we use a green fluorescent protein in recombinant DNA technology. But what you can actually need to know just to answer some of the structure question. So number 2, very important. Number 2, very important. Okay, minimal invasion means that we do not need to kill the cells because we can view it under the conventional fluorescent microscope. So number 1, number 2, you can put it together. Okay, and then number 5. Okay, and then number 5, high level. Okay, no toxicity, so that's why suitable to be used. Number 6. Okay, number 6, actually what we can do, you just focus here. GFP can be used to study the behavior of the tag protein within the living cells. Okay, so number 6, but do not need to write so long, just the last part. Okay. Yeah, focus in terms of these 4 points. Okay, but for green fluorescent protein, there are some disadvantages or limitation as well. Okay, so what are the limitations? Because they're resistant to the protease. So means that they will remain there. They will still remain there, even though the expression is stopped already. Okay, now what I mean here is, okay, let me clear up this part. Now, as I said that, I can study the expression, time point expression, time. So when I start expressions, you can see the graph continue. And then, consider. But if I want to study the degradation of a particular protein, then I can't study already. Because why? It's very stable. So it continues like this. In fact, if I stop the expression here, if the expression stop here, so the cell should remove it, right? Can I see that? But the degradation here won't be shown. Are you clear? Won't be shown already because it's very, very stable. It won't be degraded. So you can't use it to monitor the dynamic change in the gene expression. So it means up and down, you won't be able to see it. Okay, for example, we want to see up, okay, then down, express, not express. You won't be able to see this kind of the graph anymore because the green fluorescent protein is very stable. Okay. And green fluorescent protein is non-enzyme protein. Now, this is very important. Highlight number 2. Okay? Green fluorescent protein is non-enzyme protein. So if you compare to those enzyme protein, GFP have a little bit of what we call this disadvantage of limitations here. Now, let me show you what actually happens if you compare between these 2 kinds of the methods. Now, we use enzyme. Enzyme, we understand that enzyme can be reused. So they have what we call the turnover. Okay, turnover number. Okay, but GFP is protein. So proteins cannot be reused. Are you clear? And they don't have the turnover. So what does this mean? So sometimes the expressions, okay, the expression inside the cells might be too low. Try to imagine, this is the cell. So they may only produce, okay, few enzymes. Okay, let's say 4 enzymes here. They produce 4 molecules. Are you clear? So here they also produce 4 in this case. Okay, the cells also produce 4 GFP. But if you shine with UV, what will happen here is only 4 green fluorescent protein. So you may not be able, too low. The expression is too low and may not be able to pick up. So it means that it will show us a negative result. Oh, the cell would express. Can I see that? Okay, because the intensity is too low for the equipments or device to pick up. Why? Why we do have this problem? Because no turnover. You produce 4 means only 4 available. But if you use the enzymatic reactions, what will happen here is enzymatics, this enzyme, even though 4 of them only, they can help us to convert the substrate into the product. So the product will give us the detection. We can detect the product. And because one enzyme can convert 1,000 of the substrate to the product, so means that product's concentration increase until we can detect. So means that if you use a green fluorescent protein, okay, if you use green fluorescent protein, what will happen here is that is expressions, right? Cannot, we get green fluorescent protein, but the expression is too low for us to pick up the signal, and we claim that is a negative. So this negative we call as a false negative. Are you clear? False negative. So means that by right, it's a positive result, got expression, but we can't pick it up the signal, right? Because due to the sensitivity of the device is not so sensitive, therefore we won't be able to pick up the signal. So that is a limitation of the green fluorescent protein because it is a protein, not enzyme. So without, they don't have the characteristic of enzyme, they don't have the turnover number, therefore If the signal is too low, we may get false negative result, where by rights, the cells already give a certain signal, but we can't pick up the signal. But this thing won't happen if we use enzymatic reactions, because enzymes, even though low number enzyme, but because they can reuse and they can be reused, and also they have a turnover number, they can continue to convert the substrate into the product. So until the product reach a certain concentration, it becomes detectable. Are you clear? So in this case, you won't have the false negative results when you come to the enzyme. So what enzyme we can see here is the X-gal. X-gal Cronut, okay, in the X-gal, the blue white screening, where you have the beta galactosidase. When the beta galactosidase continuously converts the X-gal into the blue compound. And because of this, we can see the blue colonies in the this petri dish. Okay, so there's a special about the enzyme versus protein. So this question actually come out in your past year before. So please do aware about this. Okay, the limitations of the green fluorescent protein, because they are non-enzyme and they don't have a turnover number. So the signal may be too low to be detected by the device. Okay. Now the last one, because we can actually expose the living cells via UV, and the UV may cause the cells damage. Okay, because time point study, every hour we expose UV, every hour we expose UV. So over a long period of times, it may damage the cells. Okay, or it may cause the mutations in the cells. So this again is a limitation compared to the enzyme, the X-gal. X-gal, you don't need to use this UV, right? You don't need to use a UV, you just use a naked eye. You can see the color change already. But for the GFP, it won't give us any signal when you view it under the normal microscope or using our naked eyes. Okay. So as against what you need to know for the green fluorescent proteins, not all, generally you should know that very minimal invasion. We do not need to kill the cells in order to view the expression. Okay, and we do not need to use a very sophisticated device to check on the signal. We can use the fluorescent microscope, or even the UV lights can help us to detect already. Okay, and also high level of GFP expressions have not associated with any cellular toxicity. The problem here is not because of green fluorescent protein, but UV. Okay, UV is the one that may kill the cells, but not the green fluorescent protein. And last one, green fluorescent protein can be used to study the behavior of tag protein within the living cells, because we don't need to kill the cells, so you can see the dynamic change here. Okay. So, but the limitation here is GFP is a non-enzyme proteins. So therefore, the lack of ability to amplify its signal through the enzymatic turnover. So the order of magnitude is less than the that of the other enzyme-based reporter systems such as beta galactosidase and X-gal. Okay, so it may give us the result. Okay, positive result, it may claim as negative. Okay. But no matter how, the very important highlight point number 15, we want to reduce, okay, whether we use enzymatic system or fluorescent protein, whether in a fluorescent protein or chromogenic substrate using enzyme, we want to reduce the risk of spreading antibiotic resistant genes to the pathogenic bacteria through the horizontal gene transfer. Okay, so now let's come to the last part of this one. We have already done the screenings. The next part, we're going to clones, okay, the transform host cell. So once we confirm, okay, this colony can produce the insulins for us, then we actually, okay, use a fermenters. Or we can write it's a bioreactor. So this bioreactor or fermenters is something like a house, a chamber, where we can provide a very optimum conditions, sterile and optimum condition for the growth of the microorganism, such as the transform cells. So you can see that this is a basic fermenters, the design here. So if let's say the bacteria require the air, it can actually channel in the air, and we have the stirrer here to make sure that the temperature is even. We have the pH probe to make sure that the pH is optimum, and then we do have the cooling water, so that to make sure that it won't be overheated inside this chamber. Okay, and we do have the temperature control to make sure that, okay, the bacteria is provided with a suitable temperature. So once they reach a optimum temperature, they will continue to grow and produce the insulin for us. So we do have the harvest line here. Okay? The harvest lines can help us, right, to take out the, for example, the cells, and then we can harvest. Okay, so inoculums and nutrients, we can always pump it in. Okay, so now one thing I want to actually inform you guys, if you look at the past year paper, you can see that we do have what we call the batch culture, fed bed culture, continuous culture. So those are no longer in our syllabus. So when you do a past year, you can skip those questions. Batch culture, fed batch culture, continuous culture, okay, secondary metabolites. Too bad those are quite good topics, but have been removed from your syllabus already. So too bad you guys won't be able to learn those. Okay, those are very, very important in future if you learn or you plan to pursue your degree in biotech, because in biotech you need to culture the bacteria, you need to culture the fungus, so that it can produce certain protein for us. But now this topic have been removed from your syllabus already. Okay, so if you look at this past year paper regarding the batch culture, fed batch culture, continuous culture, secondary metabolite, penicillins, those are not in your syllabus anymore. Okay, so what actually happened inside these fermenters? So the transformed bacteria are going to undergo DNA replications, and that's why it's very important your bacteria must be able to recognize the origins of replications, the ORIs. Okay, and they carry out the binary fissions. Then it can we will be able to transfer the genes from one cell to two cells, 2 to 4, and continue. Okay, and be careful, guys. I have seen students talk about this. Bacteria, when you use the bacteria cannot carry out mitosis. Yeah, they don't have nucleus. So don't write, I mean, bacteria carry out mitosis. Okay, no. Okay, don't write this. Okay, so bacteria only will skip the mitosis. They will have the DNA replications. After DNA replication, they will skip the mitosis and they go for the cell divisions. So this cell division is by binary fissions. Okay, by binary fissions. Okay, be careful when you write. Okay, so when a recombinant plasmids, okay, contains the promoter and the right control sequence, okay, for example, the operator, then bacteria can be induced to express the foreign genes it contains when the chemical signal is added. Okay, so basically we can actually activate the genes and deactivate the gene. Okay, so that's okay. We want them to produce insulin. We give them a particular control sequence, or we give them particular chemical signal inducer to induce them to produce the insulins for us. Okay. So this is why I say that colony grow in a large culture, and then we continue until we can get the mRNA to be translated into proteins. Then we get the proteins. Okay, and we do know that promoter allow RNA polymerase to bind, and operator act as a switcher. We have learned this one in the previous chapter. So go and look at the gene control in prokaryotic cell. Sorry. Okay, gene control in prokaryotic cells, the last previous chapter. Okay, so now, because if you use the bacteria don't have the Golgi apparatus, they can't secrete the protein for us. No choice, we have to lyse the bacteria. And your concern here is, if I lyse the bacteria, then no more bacteria. So we don't do this, okay? We don't take everything out from the fermenters. So we can actually take out half. For example, we harvest half, remaining half, we still continue to give them nutrient, they will continue to grow. Can I see that? So we won't need to actually continuously to produce a new batch of the bacteria, okay, or transform bacteria. Okay, so we actually need to harvest half, and the remaining half we can we provide them with the suitable nutrients, they will continue to grow. Okay. So how we actually because bacteria not only produce insulin, bacteria can produce their own protein, proteases, enzyme as well. How we do the purification? So the purification can be done by using this affinity chromatography. Okay, not in our syllabus, just for information, because we talk about the purification. So how we do the purifications? Basically very simple. If you look at this column are packed with the antibody. So this antibody actually, if you look at this antibody can bind with the insulin. So we just pour the mixture So when the mixture pass through, so insulin will bind to the antibody, the rest of the protein will be released out from this column. Then we perform the washing, wash away all the impurities here, and then we change the pH because the binding ability of the insulins to the antibody depends on the pH. So if you change the pH, then you can see that the affinity become lower, then we can collect the insulins after that. Okay, so first we trap the insulins, after that we release the insulin. So this method is called as affinity chromatography. Okay, so affinity chromatography allows us to purify the sample. Clear? Now, something special about eukaryotic cells. If you use a eukaryotic cell as the host cell, then we need to add the transcription factor. So what is the function of transcription factor? So we have learned this one in the gene control in eukaryotic cells. So in eukaryotic cell, a bit special. If we have this of gene of interest and we have the promoter. So as I said before, RNA polymerase can bind to the promoter in the eukaryotic cell directly to initiate the transcription, but in the eukaryotic cells RNA polymerase cannot directly bind to the promoter, so we need to have the transcription factor to bind. So when the transcription factor bind already, RNA polymerase only can bind to the promoter and initiate the transcription process. Okay, so that's why if you use a eukaryotic cells, one of these advantage here, you need to know what is the transcription factor needed, and then we add in so that transcription factor will bind to the promoter region or to the RNA polymerase before the transcriptions can begin. Okay, so this figure summarizes the entire process, how we actually manage to get the human insulin production from the genetically modified bacteria. Okay, so with this, have done for the gene transfer.